نجي هسه على آخر درس اللي هو immune tolerance. Basically in this lecture we will know the principles of immune regulations, the three principles, the self-tolerance, mechanism of central self-tolerance and peripheral self-tolerance for T-cell and B-cell, and we'll talk about the inhibitory receptors of T-cells. Now the immune system must be in an immunological equilibrium. There must be a balance in the immune system. It must be activated, not too weak in order to get rid of the pathogen, and not too strong to cause tissue damage. We need to be normal where reactions against pathogens occur. However, we don't want it to be over. So we don't want an immune response against self-antigen and harmless antigen, which is tolerance. Now, the importance of immune regulation. Immune regulation is so important because number one, wait, what? بس لحظة. شلون؟ تمام. The importance of immune regulation. Number one, to prevent inappropriate reaction against self-antigen, we need self-tolerance. So no autoimmune disease. To prevent immune responses against harmless environmental antigens, like commensal microbes, and this happens to prevent allergies. And thirdly, to avoid excessive lymphocyte activation and tissue damage during normal protective responses against infection. يعني اكو pathogen and we need the immune response, but we don't want this immune response to be very strong or get out of control because it will lead to tissue damage. Now, failure of the control mechanism is the underlying cause of immune-mediated inflammatory diseases. نجي هسه على immunological tolerance. Tolerance is basically unresponsiveness to an antigen. You tolerate. Do you tolerate someone means that you do not have a problem with them. So it's unresponsiveness to an antigen. This is triggered by exposure of lymphocyte to that antigen. Now, the significance of tolerance. All individuals are tolerant of their own antigens, which is self-tolerance. Now, breakdown of self-tolerance, which it becomes self-intolerance, it will result in autoimmunity, as I mentioned. The therapeutic potential, basically inducing the tolerance, may prevent the graft rejection, treat autoimmune diseases and allergic diseases. زين هسه بالنسبة للتي cell, there are two types of tolerance. You have the central tolerance, which happens in the primary lymphoid organ اللي هو الثايمس بهاي الحالة, وعندنا peripheral tolerance اللي يصير بالsecondary lymphoid organ. راح نتكلم عن central tolerance which happens in the thymus. هسه بالنسبة لل positive and negative selection, central tolerance in the thymus, we have three outcomes. If the T cell recognizes the antigen, it might undergo apoptosis or deletion, negative selection, or the CD4 positive, the helper T cell, might develop into regulatory T lymphocytes. هنا كاتبين some T cells may differentiate into regulatory suppressor T lymphocytes اللي هي regulatory T cells. And regarding the B cells, طبعا هذا بالT cells. بالنسبة للB cells احنا قلنا انه it has a chance. The B cells has a chance in order to edit the receptor, but this does not occur in the T cells. زين central primary lymphoid organ. إذن بالthymus, for the T cells, you either, when there is recognition of a self-antigen, you either have apoptosis or development into regulatory T cells. Now, for the peripheral T cell tolerance in secondary lymphoid organs, basically in A, it shows the normal immune response or immune activation of the T cell. It requires two signals. The primary signal, which is the MHC binding to the TCR, holding the antigen, and the second signal, which is the co-stimulator the B7 binds to the CD28, it will proliferate and differentiate into effector and memory T cells. هسه بالنسبة في حالة إنه هذا الأنتيجين was a self antigen, peripheral tolerance, there are three mechanisms. The first mechanism is Anergy, the second mechanism is suppression, the third mechanism is deletion. نجي هسه على Anergy. As you can see here, we have the class MHC holding the self antigen binds to the TCR, but we do not have the co-stimulatory signal, which is the B7. So this will lead to functional inactivation or energy of the cell. The second mechanism is basically suppression, and this is with the help of regulatory T cells. هسه راح نعرف شلون تسوي يعني regulatory T cells تسوي هذا الشيء طبعا through cytokines and inhibitory receptors وإلى آخره on both the T cell and the dendritic cell. The cytokines produced by the regulatory T cells travels to the T cell, block at the activation. وبالنسبة للinhibitory receptors, for the inhibitory receptors on the regulatory T cells deals with the dendritic cell. We will explain. Deletion, basically if the T cell recognizes the self antigen, it undergoes apoptosis. طبعا كلها هاي الحالات في حالة انه الانتيجين الموجود هنا is a self antigen and it has been recognized by this T cell. Either it undergo energy or suppression or deletion. Now the T cell energy, there are two ways or two mechanisms, and I mentioned one of them, which is the signaling block. Signaling block, as you can see, we do not have the B7, the co-stimulatory signal. Now the T cell, if it recognizes the antigen that is held, the antigen which is the self antigen that is held by the class MHC, it will undergo energy, as you can see here. There's no co-stimulatory signal. Now the second way of this energy, basically by engagement of inhibitory receptors. Basically the T cell, if it recognizes the self antigen, the T cell has inhibitory receptors. These inhibitory receptors will explain later what are they, the CTLA4 and the PD1. It basically leads to energy. It prevents the activation of the T cell. Now as I mentioned هنا هذا اللي قلته انه T cell responses are induced when the cells recognize an antigen presenting by an APC activating receptors on the T cell CD28 recognize the co-stimulatory B7. If the T cell recognizes the self antigen without the co-stimulation, which is the B7 binding to the CD28, the T cell becomes unresponsive or Anergy, which I mentioned, the signaling block. Or basically engagement of inhibitory receptors such as the CTLA4 and PD1, the T cell will undergo energy, will remain viable, but will not respond to this antigen. بالنسبة للسuppression, suppression هنا done by the T regulatory cells اللي لها اسم ثاني اللي هي suppressor cells, as we mentioned. هسه regulatory T cells, there are two sources. Regulatory T cells can be formed from the primary lymphoid organ. Basically, a T cell recognizes the self antigen. As I mentioned there, it could differentiate into regulatory T cell. So this type of regulatory T cell is called the natural regulatory T cell, or NTreg. Now, if the T cell, the CD4 T cell, recognizes the antigen in the lymph node, for example, secondary lymphoid organ, some T cells will differentiate into a regulatory T cell, and this will have the name of induced iTreg, induced. Eventually, we have the regulatory T cell which have the FOXP3, the transcription factor or the marker FOXP3. It inhibits what? It inhibits the T cell activation. Not only it inhibits the T cell activation, it also suppresses the activity of the already activated T cells during normal immune responses to prevent the immune response from getting out of control. It also inhibits other cells like natural killer cells and B cells. Then the regulatory T cell are generated by self-antigen recognition in the thymus, which is natural regulatory T cell, and Treg, as I mentioned. Peripheral lymphoid organ, induced or inducible T regulatory cells. And peripheral tissue Tregs. طبعا الريجولاتوري تي سيل الاكشن هاي شغله كلش مهمه، واجا سؤال بالتي بي ال واني غلطت بيه، فحبيت يعني اقوله انه على المود يعني لا اني انساه ولا انتي تنسي، اللي هو انه الاكشن of the regulatory T cell happens in the secondary lymphoid organ in the peripheral tissue only. It doesn't occur in the primary tissue. It suppresses the activation of T cell that recognizes the self-antigen. It inhibits the already activated effector T cell during normal immune response to prevent the immune response from getting out of control. It inhibits the activated B cell and natural killer cells and suppresses the activation of self-reactive lymphocytes. Now, inhibitory receptors of the immune system. One mechanism by which the system maintains a balance between the activation and inhibition, which is immune regulation, is the use of different receptors. Now, inhibitory receptors are present where are not only present in the T cells or the Treg regulatory T cell, it is also present in the B cells and the natural killer cells. هذول ما أعرف شنو سالفتهم. المهم there are two types of inhibitory receptors, the CTLA4 and the PD1. The difference between them is that the CTLA4 works in early immune response. However, PD1 works in late immune response، يعني بنهاية الاميون response. راح نركز احنا على CTLA4، ما راح ندخل بالPD1. بالنسبة للactivation and inhibitory receptors on T cell, we have the CD28 and B7 binding that will result in the activation process of the T cells. هسه inhibition, we have the receptor, we can use the receptors. Now the CTLA4, it competes with what? It competes with the CD28 in binding to the B7 on the APC. In binding to the B7 on the APC. And it is present where? It is present on the T cells that is being activated and the regulatory T cells. وهنا it inhibits early T cell responses in the lymphoid organ. فلذلك احنا قلنا انه it works in early immune response. The PD1, it does not compete with the CD28 and it does not bind to B7. What it does, there is on the APC, antigen presenting cell, like a dendritic cell, there is a specific receptor which is PDL1 or PDL2. PD1, which is present on the T cell, binds to these PDL1 and PDL2 in order to prevent the immune response from getting out of control, to suppress the action of the effector cell. ما راح نركز عليها، حنركز على CTLA4 فقط. نجي هسه على opposing function of CD28 and CTLA4. CD28 للشنو؟ للactivation. CTLA4 للشنو؟ للinhibition. زين؟ عندنا الnormal B7 CD28 interaction, as you can see here. B7 will bind to CD28 co-stimulatory, will activate the naive T cell, and proliferation will happen first, then after that differentiation into effector and memory T cell. أما في حالة إنه كان عندنا CTLA4 B7 interaction, CTLA4 on the T cell will bind to the B7. It competes with the CD28 طبعا. And after that, it will remove this B7 from the APC and ingest it into the cell. Basically, what will happen is Anergy, T cell energy, functional inactivation. نجي هسه على mechanisms of action of Tregs. As we said, it suppresses the immune activation on both the normal immune response to resume homeostasis and suppress the lymphocyte activation to self-antigen. إنه يعني مو بس it inhibits the activation of T cell when it recognizes the self antigen, it also, when there is a pathogen, normal immune response, the T cells will be activated to the pathogenic antigen. After the recognition of pathogenic antigen, it would suppress the effector T cell in order to prevent the immune response from getting out of control. CTLA4 on the Treg removes the B7 on the APC, which reduces the CD28 engagement and T cell activation. طبعا هنا CTLA4 competitively inhibits B7 and CD28 engagement. هنا ال co-stimulation will lead to T cell activation. لكن لو كان عندنا مثلا CTLA4 will bind to the B7, competes with the CD28. It has higher affinity to the B7, binds to it, removes it from the APC and ingests it. طبعا احنا لما يكون عندنا ال APC احنا بس نوضح انه اكو هناك بس B7 واحد. طبعا يكون اكو هناك اكثر من B7. So CTLA4 will reduce the number of B7 receptors. So as a result, B7 will be reduced, so activation will not happen and energy will occur. Now CTLA4 is an endocytic receptor. What does it mean by endocytic receptor? When it binds to the B7, it enhances endocytosis of the B7 receptor that will ingest the B7. And it will remove and ingest these molecules. When CTLA4 is expressed on either Tregs or activated T cells, it outcompetes the CD28 and reduces the amount of B7 available on the APC, reduces the number and the amount. And كلام دي ينعاد هنا دي يوضح انه CTLA4 has much higher affinity for B7 than CD28 والى اخره. زين هسه CTLA4 there are two يعني actions. You have cell intrinsic action and you have cell extrinsic action. The cell intrinsic action لما CTLA4 is present on the T cell that is being activated اللي هي responding T cell. زين في حالة انه كان الانتيجين هنا self antigen and this T cell has recognized this self antigen وفرصة معينة كان اكو co-stimulatory B7 co-stimulatory B7. هسه اللي راح يصير انه ال CTLA4 on the T cell will compete with the CD28 in binding with the B7, binds to the B7, removes it from the APC, ingests it, and prevent the activation of the T cell against the self antigen. وهاي cell intrinsic. يعني cell intrinsic like يعني مثل انه T cell has inhibited itself. الcell extrinsic هنا T regulatory cells تتدخل. احنا مو قلنا انه CTLA4 can be present also on the T regulatory cells. فالregulatory T cell it has the CTLA4, it binds to the B7, ingests it, removes it, reduce the number of B7 receptors on the APC. وبالتالي it will number one prevent the activation of the T cell against self antigen. Number two ممكن هاي responding T cell is already activated an effector T cell to a specific pathogen in normal immune response, and this regulatory T cell has suppressed and limited the action of this effector T cell in order to prevent the immune response from getting out of control. تلاحظين قاعد أعيد الكلام أكثر من مرة لأنه كلها مرتبطة ببعض. وهنا مكتوب هذا الكلام إنه this happens as normal mechanism of suppressing normal immune responses إنه في حالة إنه infection أكو or a process to suppress the activation of self antigen of T cell recognizing self antigen. هنا مرة ثانية دي عاد limits the activation CTLA4 limits the activation of the responding T cell, mediates the suppressive function of regulatory T cells. Now السؤال اللي يطرح نفسه شلون التي سيل تختار انه هي هسه عندها CD28 and CTLA4 شلون تختار منو اللي يرتبط بالبي 7. It depends on two things. It depends on the affinity and it depends on the expression of the B7. في حالة إنه ال APC اللي هي ال dendritic cell had no B7 or low B7. شنو معنى هذا الكلام؟ معنى هذا الكلام إنه there is no pathogen and the antigen presented on the class MHC is self-antigen. فإحنا منريد CD28 engagement in order to bind to the B7. So basically what will happen, engagement of CTLA4 binds to any B7 if there are, to prevent the immune activation against the self-antigen. أما في حالة إنه كان أكو عندنا high B7 means there is a pathogen inside inside the dendritic cell and the antigen on the MHC is pathogenic. فبهذه الحالة احنا نريد activation of the T cell. فهنا نختار ال engagement of CD28 in order to be activated وإلى آخره. نجي هسه آخر شيء بالنسبة للبي cells. There are two tolerances, central tolerance and peripheral tolerance. لكن أكو بعض الاختلافات. احنا قلنا إنه البي cell if it recognizes the self antigen it has a chance in receptor editing. Receptor editing, basically new rearrangement of the light chain because it is easier to change. Okay? هسه it does the receptor editing. Now if it recognizes the self antigen again, it will undergo apoptosis. زين? Or deletion. هسه بحالة central tolerance we have high avidity self antigen recognition and low avidity self antigen recognition. Avidity هي نفسها تقريبا affinity. في حالة إنه البي self reactive B cell there is a high avidity self antigen recognition strong. إذا لاحظتي هنا بالرسمة the receptor, the B cell receptor like fits the antigen, the self antigen. فهذا strong affinity. It might undergo receptor editing in order to like give it a new chance and produce non-self-reactive B cell, or it might undergo apoptosis. Maybe it is the second chance. There is no second chance. يعني هي فرصة واحدة وبس. Deletion. أما في حالة إنه كان عندنا recognition of the self antigen but low avidity, low affinity. حتى لو لاحظتي هنا الريسبتور doesn't completely fit. لاحظي self antigen is like a square and the receptor is like a circle, so it doesn't like completely fit. So low avidity or low affinity would lead to reduced receptor expression. لاحظي receptors كان اكو 3 هنا صار اكو 1 والسيجنالينج يقل. It will cause energy of the B cells. وهذا أول اختلاف بين البي cells والتي cells. التي cells الانرجي وين يصير؟ بالperipheral tissue. البي cell الانرجي وين يصير؟ بالcentral tissue, central lymphoid organ. وهاي ممكن يسألون عليها بالامتحان. Central tolerance, immature B cells that recognize self-antigen in bone marrow with high affinity die by apoptosis or change, as I mentioned. Weak recognition, low avidity or weak recognition of self-antigen in bone marrow lead to energy, functional inactivation. نجي هسه على peripheral tolerance of B cells. 3 things might happen. If the B cell recognizes the self-antigen in the peripheral tissue, it might undergo apoptosis or deletion. It might go anergy, or also we mentioned that the inhibitory receptors are present in the natural killer cells and B cells. Inhibitory receptors basically will regulate this function and inhibit the B cells. So basically in the B cells, energy can happen in both the central and the peripheral. A T cell, but primarily it happens in the central. A T cell, it happens in the peripheral only. The peripheral tolerance in B cells, مثل ما ذكرنا, B cells that encounter self-antigen peripheral tissue can be anergic or die by apoptosis, or in some situation recognition of self-antigen may trigger inhibitory receptors that prevents B cell activation. وهنا خلصنا ال 5 lessons of immunology. إن شاء الله تكوني استفدتي.